All right, got a biology exam coming up? Freaking out a little bit about cloning? Hey, don't worry. You've come to the right place. This is your 5-minute crash course, and we're going to cover everything you need to know to absolutely crush those exam questions.
We've all seen images like this, right? In movies, on magazine covers. The whole idea of human cloning feels like something straight out of sci-fi. It's kind of fascinating and maybe a little creepy. But for your test, you need to separate that fiction from the fact. So what's the real science behind it all?
Okay, let's start with the absolute foundation. This is the bedrock. If there's one thing you absolutely have to know, it's the core definition of what cloning actually is.
So let's break this down. Cloning is simply the process of making a new organism that is a perfect genetic copy of another. The two phrases you really got to remember here are genetically identical. That means the exact same DNA, and somatic cell. That's just a fancy science term for any regular body cell, like a skin cell or a liver cell. Basically anything that isn't a sperm or an egg.
Okay, so that's the what. Now, how is it actually done? Well, the scientific name is a bit of a mouthful, somatic cell nuclear transfer. But don't let that name scare you. We're going to break it down into simple steps that are super easy to remember.
So this diagram right here, it shows you the whole shebang from start to finish. You can see you've got a tissue donor, an egg donor, and a surrogate mother. Yeah, it looks like a lot's going on, but let's just walk through it piece by piece.
All right, here we go. 6 simple steps. Step 1, you take a regular body cell from the animal you want to clone. 2, you grab an egg cell from a different animal. 3, and this is the key part, you take the nucleus, which holds all the DNA, out of that egg cell, so it's totally empty. 4, you take the nucleus from the first animal's body cell and pop it into that empty egg. It's like taking the instruction manual out of one cell and putting it into an empty factory. After that, the new embryo grows for about a week, and then finally, it's put into a surrogate mother to be born.
And here's the payoff. The baby animal that's eventually born is a clone. It has the exact same DNA as the animal that donated the original body cell. Not the egg donor, not the surrogate mother. That's the whole point of the process.
So, we know what cloning is and we know how it works. But to get that top grade, you really need to know why scientists would even do this. Let's dig into the potential applications that make this science so important.
There are 4 big potential benefits you should probably know. First, you could possibly save endangered species from going extinct forever. Second, you could quickly make copies of the best livestock to boost the food supply. Third, and this is a big one for medicine, you could create cloned organs that are a perfect genetic match for a patient, which means no more transplant rejection. And finally, you could make perfect copies of really special, genetically engineered organisms. And this right here is a perfect example of that last point. So imagine scientists go through all the trouble of creating one very special goat, a goat that can produce life-saving medicine right in its milk. That's a huge deal. So how do you make more of them? You don't start from scratch. No way. You just clone the first one. It's a way to reliably copy an incredibly valuable animal. But, and this is a huge but, no discussion of cloning is complete without looking at the other side of the coin. A really good exam answer always talks about the big problems and the ethical debates. So let's dive into those counterarguments. And the downsides, well, they're pretty serious. You have massive ethical, moral, and religious objections right off the bat. Then there's the practical side. The whole process is unbelievably expensive, and it has a very, very low success rate. A lot of clones fail to develop or are born with major health problems. And then there's the ecological risk. Cloning reduces biodiversity. If you have a whole herd of animals that are all genetic copies, one single disease could come along and wipe them all out in one go. So, this really lays it all out, side by side. On the one hand, you've got this incredible potential for medical miracles, for feeding the planet, for saving species from the brink. But on the other hand, you have the reality of sky-high costs, frequent failures, deep ethical questions, and a real risk to genetic diversity. It's a massive debate. Phew, okay, we've covered a ton of ground. So, let's boil it all down. What do you absolutely, positively need to have locked in your brain for the test? Let's go over the 4 key essentials. All right, here they are, your 4 key takeaways. Number 1, cloning makes a genetic twin from a regular old body cell. 2, the main technique is called somatic cell nuclear transfer, or just SCNT for short. 3, the biggest potential benefits are in medicine, farming, and conservation. And 4, the major problems are the crazy high cost, the low success rate, and of course, the serious ethical issues. You got that? Good.
So I'll leave you with this final thought. Perfect for an essay question, by the way.
We have this incredibly powerful technology that could save species or grow life-saving organs. But when you factor in the huge cost, the high rate of failure, and the ethical lines we might be crossing, the big question is it all worth the risk? Definitely something to think about. Good luck on your exam!